Slide 1. This slide tape is the third in a series of presentations in acid-base physiology prepared by the American Physiological Society. 2. This program begins with a review of prerequisite knowledge. Before you go any further, make sure you have completed the entry skills test provided in the study guide. 3. During the presentation, question slides will appear at intervals. Stop the tape to answer the question. Then advance to the next slide on which the correct answer will appear. Stop the tape again to check your answer. After you finish the unit, assess your understanding of the material by taking the post-test in the study guide. 4. This unit discusses the role of hemoglobin in hydrogen ion metabolism. Content expert, Dr. Arthur Otis. 5. We will briefly examine those properties of hemoglobin which determine its role in hydrogen ion metabolism. First, reversible combination with oxygen. Second, reversible combination with carbon dioxide. Third, buffering capacity. 6. As your first objective, you should be able to 1. Draw a curve showing the reversible combination of hemoglobin with oxygen. 2. Define the Bohr effect. And 3. State the condition necessary for, first, the addition of carbon dioxide to hemoglobin to form carbamino compound, and second, for the release of carbon dioxide from carbamino compound. 7. Hemoglobin can combine reversibly with oxygen. This property is a nonlinear function of the PO2. Blood leaving the lungs normally has a PO2 of 100 millimeters of mercury, and hemoglobin is nearly fully saturated. The mixed venous blood coming from the various tissues has a PO2 of about 40 millimeters of mercury and is 75% saturated. 8. A change in PCO2 or hydrogen ion concentration alters the affinity of hemoglobin for oxygen. This phenomenon is called the Bohr effect. For example, at a constant PO2 of 50 millimeters of mercury, a PCO2 of 20 millimeters of mercury results in approximately 90% saturation, while a PCO2 of 80 millimeters of mercury results in approximately 75% saturation. 9. Metabolism adds CO2 to the blood as it flows through the tissues. Thus, the acidity of the blood tends to increase. Due to the Bohr effect, the affinity of hemoglobin for oxygen decreases, causing oxyhemoglobin to dissociate. As a consequence, the availability of oxygen to the tissues is increased. 10. In the lungs, the reverse takes place. As PCO2 decreases, the hydrogen ion concentration decreases, and the affinity of hemoglobin for oxygen increases. 11. Hemoglobin can also combine reversibly with carbon dioxide. This property is a function of certain amino groups in the protein moiety, which combine with the CO2 to form carbamino compound and hydrogen ions. 12. More carbamino compound can be formed by unoxygenated hemoglobin than by oxyhemoglobin. 13. Thus, the deoxygenation of blood in the tissue capillaries enhances the formation of carbamino compound. 14. While the oxygenation of blood in the lungs favors the release of CO2 from this compound. 15. Of the total CO2 carried in the blood, only about 10% is exchanged during each passage through tissues or lungs. 16. Of the 10% exchanged, about 10% is attributable to changes in dissolved CO2, 30% to changes in carbamino CO2, and 60% to changes in bicarbonate concentration. 17. Remember to stop the tape to answer questions and to check your answers. 18. 19. 20. 21. As your second objective, you should be able to 1. Identify the relative strength of hemoglobin and oxyhemoglobin as acids, and 2. Define the Haldane effect and state its consequences. 22. In addition to its ability to combine reversibly with oxygen or carbon dioxide, hemoglobin is the major protein buffer in the blood. 23. Both hemoglobin and oxyhemoglobin are weak acids, which can dissociate to form hydrogen ions and a conjugate base. 24. In accordance with the isohydric principle, these protein buffers interact with the bicarbonate buffer system and enhance its effectiveness. Both oxyhemoglobin and hemoglobin are weaker acids than carbonic acid. However, oxyhemoglobin is a somewhat stronger acid than unoxygenated hemoglobin. 25. Because oxyhemoglobin is a stronger acid than unoxygenated hemoglobin, the degree of oxygenation of hemoglobin at any given PCO2 will affect the concentration of bicarbonate. This phenomenon is called the Haldane effect. 26. Deoxygenation of hemoglobin will result in an increase in the concentration of bicarbonate and a decrease in hydrogen ion concentration. Oxygenation will result in a decrease in bicarbonate and an increase in hydrogen ion concentration. 27. Blood flowing through metabolizing tissues takes up CO2 and tends to become more acid, and in accordance with the Bohr effect, gives up more oxygen. However, as a result of the Haldane effect, the increase in unoxygenated hemoglobin tends to make the blood more alkaline. These opposing simultaneous effects almost exactly neutralize each other, so that the difference in hydrogen ion concentration between arterial and venous blood is minimized. 28. The reverse occurs in the lungs. Loss of CO2 tends to make the blood more alkaline, and the Bohr effect enhances formation of oxyhemoglobin. But because of the Haldane effect, oxygenation tends to make the blood more acid. As in the tissues, the net result is that the change in hydrogen ion concentration is minimal. 29. 30. 31. 32. 33. In summary, 1. Hemoglobin combines reversibly with oxygen. 2. Hemoglobin combines reversibly with carbon dioxide. And 3. Hemoglobin as a protein buffer enhances the effectiveness of the bicarbonate buffer system. 34. Fourth, hemoglobin, through the reciprocal action of the Bohr and Haldane effects, allows the simultaneous exchange of oxygen and carbon dioxide mutually to enhance each other, and minimizes the change in hydrogen ion concentration when gas exchange occurs both in the tissues and in the alveoli. 35. This concludes the discussion of the role of hemoglobin in hydrogen ion metabolism.